In this lesson, we will review TEKS B.11B. Investigate and explain the role of enzymes in facilitating cellular processes. These chimpanzees are having an afternoon snack. To make use of the nutrients in the fruit, the chimpanzees' digestive systems must break the fruit down into tiny particles. That job is handled by enzymes. Here we have a DNA molecule getting ready for an important process. It is about to be replicated. For replication to take place, the two DNA strands need to be separated from each other. What is this molecule that is separating the two DNA strands? Once again, it's an enzyme. Enzymes perform millions of jobs like these around the clock to help make our bodies and our cells go. You can think of enzymes as tiny molecular tools that help cells and organisms perform all kinds of life processes. Let's explore the role of enzymes in more detail. All living organisms have enzymes. Humans, for example, have approximately 75,000 different types of enzymes. Enzymes come in all kinds of shapes, and each unique shape is suited to its job. The enzymes you see here illustrate just a few of the limitless examples of enzyme shapes. So, what exactly are enzymes, and how do they do these important jobs? Enzymes are biological catalysts. A catalyst is any substance that helps speed up a chemical reaction. So, enzymes are molecules that speed up chemical reactions in living organisms. Nearly all enzymes are proteins. Proteins, which are made up of chains of amino acids, are able to fold into the vast array of shapes that make enzymes unique. Let's look at a few examples of enzymes and what they do. RNA polymerase is the enzyme that helps cells build a new strand of RNA during transcription, an important step in the process of making proteins. Lactase is a digestive enzyme. It helps an organism break down lactose, a sugar found in dairy products. Have you ever heard of lactose intolerance? It's a condition in which people have trouble digesting dairy products, and it occurs when the body doesn't produce enough lactase enzyme. Cyclin-dependent kinase is one of the most important enzymes involved in cell division. It helps regulate the cell cycle. Lipase is another kind of digestive enzyme. It helps an organism digest lipids or fats. Peptidyltransferase is an enzyme that is important for making proteins. It helps assemble amino acid chains during translation. Notice that in this table, all of the enzyme names end in the suffix -ase. In most cases, an enzyme's name refers to its function, and it ends in -ase. For example, RNA polymerase helps polymerize or build RNA. There are exceptions, though. Trypsin is the name of a digestive enzyme that breaks down proteins. These are just a few examples of enzymes and their impacts on life processes. But how exactly do enzymes help carry out these life processes? As mentioned earlier, enzymes are catalysts, which means they speed up chemical reactions. We'll use this chemical reaction as our model. In this reaction, a reactant is being broken up into 3 products. When the right enzyme is present, this chemical reaction can take place more quickly. Let's illustrate this on an energy graph. An energy graph shows us how much energy is needed to complete a chemical reaction. In this case, this red arrow represents the amount of energy it takes to complete our chemical reaction when there is no enzyme around to help. And this blue arrow represents the amount of energy it takes to complete the same chemical reaction when the enzyme is present. Notice that the amount of activation energy needed when there is no enzyme is much greater than the amount of activation energy needed when the enzyme is present. What does this mean? It means the enzyme reduces the amount of energy needed to complete the reaction. As a result, the reaction happens more quickly. Let's take a closer look at how this works. In this chemical reaction, we have one reactant molecule breaking up into two products. So, how does an enzyme help? Here is our enzyme. This enzyme is going to bind with our reactant. So, we refer to the reactant as the substrate. The substrate is the molecule that an enzyme binds with and acts upon. The specific place on the enzyme where the substrate will bind is called the active site. The binding of the enzyme with the substrate makes it easier for the chemical bonds to form or break. In this case, the enzyme helps break the bond that holds the substrate together. Once the bond breaks, our two products are formed. It's important to note that the enzyme is not like a reactant. In other words, the enzyme is not used up in the reaction. It can be reused repeatedly to catalyze additional reactions. This is one of the many reasons why enzymes are so useful. A single enzyme molecule can catalyze millions of instances of a reaction. In this example, you may have noticed that the enzyme and the substrate seem to fit together like puzzle pieces. Let's explore that concept next. You're watching an animation of a substrate binding the active site of an enzyme. Just as in the previous example, the enzyme and the substrate have shapes that allow them to fit together. This illustrates an important concept. The shape of an enzyme and the shape of its substrate are highly specific to one another. Can any enzyme bind with any substrate? No. Enzymes can only bind with substrates that fit their active site. In fact, a typical enzyme can catalyze only one chemical reaction. No wonder there are so many different enzymes with so many different shapes. Scientists have proposed two different models to describe the complementary shapes between enzymes and their substrates: the lock and key model and the induced fit model. The lock and key model suggests that the enzyme's active site has a rigid shape and the substrate fits into the active site like a key fits into a lock. The induced fit model suggests that the enzyme's active site is somewhat flexible, and when the substrate begins to enter the active site changes shape slightly for a tight fit. Which model is correct? Both models are useful for understanding enzyme-substrate relationships, but the induced fit model is considered the most accurate. Now we know how enzymes work and the kinds of jobs they do. But is there ever a time when enzymes can't do their jobs? Yes. Enzymes are sensitive to environmental conditions such as temperature, pH, and the presence of an inhibitor. If the conditions aren't suitable, then the enzyme can't work properly. Let's start by exploring the effects of temperature. How does temperature impact an enzyme's performance? This graph illustrates the relationship between temperature and enzyme activity. Imagine that we're studying a hypothetical enzyme. We find that as the temperature rises, the enzyme's activity level increases until the temperature reaches 40 degrees Celsius. When the temperature rises past 40 degrees Celsius, the enzyme's activity begins to decline. So, 40 degrees Celsius is this enzyme's optimal temperature. That is, the temperature at which the enzyme is most active in catalyzing reactions. What happens at very high temperatures? As temperatures continue to rise, the heat begins to deform the enzyme's structure. Remember that the structure of an enzyme is very important. If it changes, the enzyme can't bind with its substrate properly, which means it can't do its job. Next, let's examine how pH affects enzyme activity. pH is the measure of how acidic or basic an environment is. A pH of 0 is the most acidic environment, a pH of 14 is the most basic environment, and a pH of 7 is neutral. This graph illustrates how enzyme activity is affected by pH. Just as with temperature, enzymes have an optimal pH. For most enzymes, the optimal pH is around 7, or neutral conditions. But think about the enzymes that do the job of digesting food in your stomach. Enzymes such as pepsin, shown here, which helps digest proteins. The stomach is a very acidic environment. So, what do you think the optimal pH of enzymes like pepsin would be? As you might have guessed, enzymes like pepsin do their best work at low pH values, around 2. Some enzymes can be impacted by inhibitors. An inhibitor is a molecule that binds with an enzyme and, in doing so, disrupts the enzyme's ability to bind with its substrate. This animation shows us an example of how an inhibitor can work. When the inhibitor, the yellow molecule, binds with the enzyme, it causes the enzyme's shape to change slightly. As a result, the substrate is unable to bind with the enzyme. Did you know that the antibiotic drug penicillin is an inhibitor? It inhibits an enzyme that bacteria use to build and maintain their cell walls. Penicillin is a different kind of inhibitor than the one we just saw. It's an inhibitor that binds directly with the enzyme's active site. When this happens, the enzyme can't bind with its usual substrate. In other words, the enzyme can't do its job. In this way, penicillin breaks down the cell walls of bacteria. Now you know how factors such as temperature, pH, and inhibitors can impact enzyme activity. As we have seen, enzymes are fascinating molecules that perform all kinds of jobs, almost like a set of tools that make our bodies and cells go. The next time you're thinking about eating a meal, replicating your DNA, or any of your millions of life processes, don't forget the role of enzymes. Let's review the vocabulary of this lesson. A catalyst is a substance that increases the rate of a chemical reaction. An enzyme is a biological catalyst, typically a protein, that binds with a substrate to speed up a chemical reaction. A substrate is a molecule that an enzyme binds with and acts upon. The active site of an enzyme is the specific location on the enzyme where the substrate binds. Activation energy is the energy input required to allow a particular chemical reaction to take place. In the lock and key model, the active site of an enzyme is described as having a rigid shape, and the substrate fits into the active site like a key fits into a lock. In the induced fit model, the active site of an enzyme is described as changing shape slightly as the substrate binds, which allows a tight fit. An inhibitor is a molecule that binds with an enzyme and disrupts the enzyme's ability to bind with its substrate. In this lesson, we reviewed TEKS B.11B. Investigate and explain the role of enzymes in facilitating cellular processes.